On behalf of Cambridge Health Tech Institute's Global Web Symposia series and our sponsor, Halo Labs, I'd like to welcome you to Finding the Needle in a Haystack, rapid assays to detect degraded polysorbate and other problems lurking in your formulation using Aura. My name is Elizabeth Lamb, and I'm the host and technical director for today's event. Now I would like to introduce our moderator for today. He is Stephen Lee, Director of Marketing at Halo Labs. Welcome, Stephen. The webinar is yours.
Hello, everyone, and thank you, Elizabeth. Good morning to everyone in the States, and good afternoon to everyone in Europe, and thank you for attending today's webinar with us. I'd like to introduce Dr. Bernardo Cordovez. He's the co-founder of Halo Labs and also our Chief Science Officer. He not only leads our R&D team, but our application efforts as well. He hails from Cornell University, earning his master's and PhD in mechanical engineering there, and he also spent some time as a postdoc as well. He's a true inventor at heart and has an illustrious career studying and developing technologies in particle analysis space, drug delivery devices, microfluidics, nanophotonics, and bioenergy production. Without further ado, the stage is yours, Bernardo.
Thanks so much for the kind intro, Stephen, and welcome everybody. Very excited to tell you how the Aura can help you quickly characterize and understand your degraded polysorbate problems in lieu of working with real monoclonal antibody or biologic formulations. So with that, let's take it away. So Aura is all about measuring aggregates and particulate impurities. At the heart of it, Aura images every single particle in your formulation. It counts them, it sizes them, and it tells you what they are using low to large volumes and in very high throughput. Now, at the heart of this webinar, I'm going to show you how the Aura can help you quantify degraded polysorbate components, these fatty acid particles, in protein mixtures, and be able to independently resolve degraded polysorbate signals from your protein particles with very high specificity and very high throughput, and also have the pictures at the end of the day to essentially help you verify these results. So this is where we're going to end up. But first, I'm going to show you step by step how do we get there. So at the heart of what the Aura measures is subvisible particles. And to my surprise, when I was essentially inventing the technology, I was very amazed at how prevalent large degraded biologic components are, whether they're subvisible or visible protein aggregates, as you see in the left panel, to AAV aggregates and gene therapy aggregates due to process instabilities, particularly very prevalent for these newer types of drugs, and even for the newer cell therapies in which cells can clump and form large aggregates as well. But the particles can either be extrinsic or intrinsic to the process, right? So it's either stability issues or it's some foreign particles that make their way into the process. But you still have to know very quickly what they are so that you can figure out whether you have a stability issue, what kind of stability issue do you have, or is it a foreign impurity. And subvisibles are incredibly important. They are a critical quality attribute. And they're actually, particles in general are the #1 cause for product recall. They present a safety concern. They've been found to be immunogenic in some cases, and in the worst cases result in capillary occlusion, resulting in even anaphylaxis. So more commonly, they're tied to reduced drug efficacy, the presence of large particulate impurities, particularly protein aggregates, as indicative of low shelf life stability. And in this case, these subvisible particles, which are again micrometer scale particles, about 1/100th of a human hair and larger, are regulated by the FDA, and there's very well USP-inspired guidelines for measuring particles larger than 10 micrometers in size. Now, many of you probably come from different backgrounds. Some of you may have actually seen a chart that looks like this, but we focus on measuring large particles larger than one micrometer and above in size. There's other techniques that are designed to measure smaller particles, including, for example, dynamic light scattering, SEC, etc. But just for orientation, we're in the same size range as the light obscuration and flow imaging techniques, and potentially image larger than those, but in higher contrast, as I'll also walk you through as we move forward. But one of the things that, one of the questions that we get pretty often is, hey, I ran my measurements, I looked at my SEC signals, for example, And I found that when I did my stress tests that these signals didn't change, meaning that I don't have small particles. Therefore, I can't possibly have large particles. But it's now very well corroborated in the scientific literature that just because you don't see small particles that you don't have large ones. These sizing domains are, in a way, largely independent, and they need to be measured as per the USP requirements. And before, it was very difficult to conduct those measurements at the earliest of stages, given the volume requirements, and that was one of the first inspirations for this whole product line. Now, how does the Aura count and size particles? So Aura enables low volume, high throughput, subvisible particle analysis in minutes. And it works with an instrument and the associated consumable. The consumable is this filter plate here that you see on the left. It has a metal frame, and stretched within it is a polycarbonate membrane. So it's a filter plate. And these black rings that you see here, these are our wells. These define the 96 independent test sites, and these are not physical wells. These are hydrophobic wells. And if you look at sample 2, potentially you can see how you can pipette 96 individual samples. But the process is very easy. First, you want to make sure that you don't consider it is a membrane microscopy technique. It is essentially inspired by US 788, but brought to the 21st century. And let me show you how it works. So the first thing that we do is we take this plate and we put it in the instrument, and we conduct a pixel by pixel backgrounding, because we do not want to confuse the texture of the membrane with the actual particle in the sample. And you can see this process is fairly invisible to the user, but you can see lines from the membrane processing and potentially some pores as well. This is a pretty clean membrane, but then this image for every well that you select is stored in memory. The next step is that you pipette however many samples of interest, say 40 microliters in replicates of 4, or an entire 96 well plate, which will span at 40 microliters a well, that's about 5 milliliters of sample. You then vacuum the plate, and you dry the samples following the USP 788 protocol, and then you reinsert this filtered sample, which now has the filtrate on it, back into the system and remeasure. Now you can see that we have these big, in this case, subvisible protein aggregates, and but there's also this texture of the membrane, right? So you could imagine how doing image analysis on something like this to say, hey, these are the particles from my sample versus these are just false positive membrane particles. It would be very difficult to do, but because we have that original image, we basically align those two with a proprietary process of image registration, and we perform a pixel by pixel background subtraction. And one of the key benefits of this is that because biologics, they're designed to live in water, because of that, they tend to have very similar optical properties of water, and therefore they have low contrast when measured with fluid imaging or other kinds of techniques. And I'll show you how we circumvent that problem. But we get these very nice high contrast images, enabling us to perform accurate detection on all biologics. Now, at first level, we produce images. The Aura produces images for every single one of the particles across all of your samples. So there's individual images, and within each of those images, their sizing, shape, and morphological parameters are characterized. So if you try to turn this particle into a circle, that's the equivalent circular diameter, to how much it protrudes out of plane using auxiliary light sources, to what is its shape. So we have that for every single particle. If we go to the well level, right, for every single well, you're going to have a particle size distribution. And this is probably one of the most popular plots in particle analysis, which is how many small particles do I have and how many big particles are present in your sample. And typically in biologics, you'll see a distribution that follows this sort of exponentially decaying curve shape, in which you have lots of small particles and ideally very few big particles. Because again, if the human capillaries can be as thin as 5 to 10 microns, you really don't want to have any big particles there. All the way to the fact that this is a 96 well high throughput experiment. So you can group all of that data and obtain high level insights. In this case, you get to see how there's 70,000 particles per mil of human IgG particles, about 40,000 particles of this protein plastic mimic called ETFE, and the negative controls hold. So again, we went from single particles to particle size distributions to the whole experiment. So we can go from very granular to high level details. And it's all very fast, meaning that you can use this, and one of the common uses has been for physical stability screening. So you can run a variety of biologic conditions under different buffer and excipient conditions, and then just characterize which one of those are most physically stable. In the case, for example, here we have 3 proteins, BSA, lysozyme, and IgG, and you can see how the BSA aggregates, they didn't produce a lot of particles. The lysozyme produced lots of particles, but actually produced very few when in this particular buffer B3, while human IgG produced the least amount of particles in this buffer B10. So this is the kind of test that would take weeks to months on traditional subvisible particles analyzers, and you can basically do this in just a few hours. Now, how do we compare against other subvisible techniques that you may have heard of or you're currently working with? So first, we are incredibly linear at low volumes. So this is an example of a serial dilution of human IgG aggregates using wells as low as 5 or 10 microliters. So if your sample is very precious, say you have a gene therapy or you're just at the earliest stages of trying to understand the physical stability of your sample, this is a great alternative. And actually, the linearity, the Count's linearity is really high. And over 3 million particles per mil can be measured linearly in the system. And one reason for that is that even though it's not intuitive, in a 5 or 6 millimeter well diameter that each of these wells has, you can really fit a lot of particles before they start stacking against each other, just from the design of the wells and enabling laminar flow, so particles want to basically land far away from each other. So high Count's linearity, and very importantly, and designed for purpose for measuring biologics. BMI is powered by high refractive index measurements. If you look at flow imaging or light obscuration, right, you're basically trying to capture the contrast between these aggregates in water, right, or these biological particles in water. And the contrast is very low, meaning the ability for a system to differentiate between black and white pixels is completely compromised. Because we conduct our measurements in air, we have very high refractive index contrast, enable us to accurately pick up even the dimmest of protein or biological aggregates. And that's been shown in the literature. Here's a paper published by Coriolis in which we were compared against 2 of the most well-known flow imagers, and we can see how, in our case, the pictures for either the monoclonal antibodies, these protein plastic mimic made by NIST, or even fatty acid particles, we have much higher contrast, meaning the difference between the black and the white pixels are much better, and therefore enabling better analysis. So that's what refractive index in action means. And in practice, typically what it means is that when compared to techniques like flow imaging, we will count more particles just again because our contrast is higher. But in general, I mean, for most samples, I think the trends are about the same, right? So we're measuring the same sample. This can vary, but for the most part, you can see that both types of technologies trend the same. We're just more sensitive and more built for purpose for measuring these very dim biologic aggregates. But one clear place, but in addition to having high refractive index contrast where we outperform, is the fact that flow imaging techniques, they work in microfluidic systems. And I'm a technologist, and I'm a fan of microfluidic systems as much as anybody. However, when you have a 3 micron tall flow cell and your depth of field is only 55 microns, you're only properly sampling about 20% of the samples that are in that channel. And this gets exacerbated, the higher magnification you get. And so, for example, there's well-established papers by Lanza for cell therapies, right, how light obscuration or even flow imaging miss more than half of the samples, right, because of this effect. Now, in our case, we don't have to worry about this. All the samples are measured in high contrast. The entire well is imaged. You count all of your sample, and 100% of your samples are on the imaging plane, right? So that basically makes sure that you're not missing particles that are above or below where your focal point is. And that's why we enable absolute particle counts. And in our newer version of technology, the Aura, we enable 100% sampling efficiency, which is very important for USP 788 requirements and for having more confidence in your measurements. Now, everything that I've discussed so far has been regarding counting and high contrast, high refractive index contrast counting. What about high throughput particle identification? At the heart of the question is, am I looking at my biological degrading, or which of my components are degrading, or or is there something else going on in my system? And it's very difficult to achieve that in the black and white world. So let me try to give this a little bit of color, inspired by our proprietary technique, fluorescence membrane microscopy. So this is these are images from a famous paper from the Japanese Pharmacopoeia, in which they were helping the U.S. NIST to essentially characterize and qualify ETFE, which are these ethylene tetrafluoroethylene particles designed to mimic the shape and refractive index and morphology of protein aggregates. And they did a pretty good job. Essentially, in these three sets of images, two of these images are actually protein aggregates. Not intuitively, these are the protein aggregates. I would have thought that the top two would have been protein aggregates, but because they look more similar. So if you resort to morphology alone, even if you have a very nice machine learning model and try to train all of your data sets, right, something like this just wouldn't work. So morphology and intensity filters by themselves, even with machine learning, it's just simply not enough to differentiate. But we've been essentially enabling all of these really great technologies that allow us that are chemically specific. One of which is not exactly Raman or FTIR spectroscopy. Now, I built Raman systems in my life, and I think the technology is really interesting. But in terms of this application, it can only be used forensically. So let's say that you were trying to identify the presence of protein aggregates or something else. First, you would need to find the particle of interest, one at a time. Then clean up, acquire a Raman spectra, look at that Raman spectra, clean it up, limit your libraries, and look for a particular peak. And if you see 1550, you might think, hey, I got an Mi2 bond. I'm looking at a protein. But the reality is that this is an incredibly time-consuming process, even with those with very trimmed material libraries. And you're basically relying on these very weak signals. And it just really takes a lot of time because you have to integrate it and it's one particle at a time. So of course it's only going to be used forensically and not really to design problems when they happen. Because ideally you want to figure out all of these problems, whether the protein aggregates, degraded polysorbate, as early as you possibly can. What we do is fluorescence membrane microscopy. And it follows the same basic 3 steps of BMI that I showed you before. You background a plate, you filter your sample, then you measure. So now you have all of your images, your particle size distributions, and all your high-level data as pertaining to all your counts. Once you've characterized that sample, you apply a dye of interest to ask a chemical question, similar to flow cytometry. How many particles are positive for fluorescence events? So if you use a protein dye, protein particles are going to show a stronger signal. If you use a lipid dye, fatty acid particles are going to show a lipid signal. If you did antibody labeling or whichever acid, those particles are going to be selected for in fluorescent glow, and then we measure them in fluorescence. So we only use the fluorescence to do chemical identification. We do not use it for sizing. But it's very powerful. And what you can now see is that with these plates that support labeled fluorescence workflows, that controls hold when applying any kind of dye. In this case, these human IgG aggregates, they've been stained with thioflavin T, which is one of the most well-established dyes for analyzing protein aggregates and inspired by decades of research in Alzheimer's. And here we have these ETFE particles that you might remember from my prior images. And in this case, they look blue, and it's the blue color doesn't mean fluorescence. It actually is from our SIMI channel, and I briefly mentioned that we can use our proprietary dark field source to determine whether particles are protruding out of plane. And here we see a mixture of the two. So not only do these particles not fluoresce, they're actually protruding out of plane, while these particles are fluorescing and not protruding out of plane. But all of that data, all of the images, is essentially captured in high throughput and can be plotted against itself or against a variety of parameters. Let me walk you through this experiment. So in this case, we have protein fluorescence channel intensity in the Y and size in the X. And each one of these dots is a particle. So the higher it is on this chart, the more strongly it fluoresces, and the lower it is, the darker it is, the more similar it is to the membrane background. So you can see that there's 2 populations, right? A population below that does not fluoresce, and you can get some really big particles that don't fluoresce, and then this sort of cloud of all of these particles that are basically potentially becoming more fluorescent the larger they get. By applying some thresholding criteria that's built into the particle view software, you can very intuitively see that and properly identify almost 100% of the human IgG aggregates fluoresced under the protein dye, while the ETFE was only called proteins less than 2% of the time. And this was done in a minute. And if you try to basically conduct spectroscopic analysis of each one of these particles, I'm not exaggerating when for a 96-well plate, it would probably take you close to a human lifetime. So how do we use this technology, fluorescence membrane microscopy, to characterize polysorbate degradation? Now, polysorbate, as most of you here will know much better than me, it's an FDA-approved excipient to increase biologic stability. It's in over 70 to 80% of all approved biologics, whether it's polysorbate 20 or 80. And it contains complex mixtures of hydrophilic and lipophilic moieties. And they've been well known to chemically degrade and enzymatically degrade and experience oxidation and hydrolysis. More simply, if you leave a biologic in the fridge or even the freezer for long periods of time, even though you should expect that because you've reduced the temperature that you're basically going to keep aggregation or instability from happening, this oxidation polysorbate degradation can still occur, and you can form fatty acid particles. So degradation results in subvisible to visible fatty acid particle formation, and one of the challenges has been that they're morphologically not too dissimilar from protein aggregates. So how do we characterize this? So not only, as you can see, these are complex mixtures, fairly, of course, proprietary, and their degradation, as I've come to learn, and many of you here know, everything in this field is sample dependent, right? So what does polysorbate degradation depend on? It depends on its composition, on the biologic sample type that is being used, on the excipients that are being used, and other materials present, as well as enzymes, refrigeration and storage, stress, you name it. So basically, it's a complex problem, and to basically cater and completely understand a complicated problem, you need the ability to conduct high throughput experiments and tackle this problem from all sorts of angles in one shot in very little time, and that's exactly what the Aura is for. So the workflow for detecting fatty acid particle degradation is very similar to the FMM workflow that I discussed before. The first off, you background a plate, you load your sample, and then you measure. You characterize all of the counts that are there, all of your particles, all of your images. Then you add one of the best established dyes for staining lipophilic particles, which is BODP FL16, part number D3821 from Thermo. And then you re-image using fluorescence membrane microscopy. I'll show you some of the insights that you can get with this technique once you do this. So to first order, let's take a deep dive on the different kinds of fatty acid particles that can be formed. Looking at polysorbate 20, some of the most common degradation elements, giving the amount of the different mixtures, be lauric acid, myristic acid, and palmitic acid. And we can see that the different fatty acids can be morphologically different, with lauric acid forming larger granules, myristic acid potentially more transparent and smaller particles, and this palmitic acid form fibrils. Now, palmitic acid can form particles that look like lauric acid, but that was just the case for this particular formulation. So, but it is to say that different fatty acid particles can look different. Now, because this problem, as you can see, starts getting a little bit more complicated, let's just look at how degraded fatty acid particles made from lauric, myristic, and palmitic acid look. So we followed a protocol designed by Doshi at Genentech et al. And we have these particles here that were basically essentially meant they were left to degrade in the fridge for an extended period of time, so basically inducing oxidation and polysorbate degradation. And then these fatty acid particles were recovered and then imaged in the Aura. And you can see this fatty acid mixture here, when it's stained with BODP D3821, we get quite strong and really pretty fluorescent signals, right? So fluorescent intensity far above the background. So not only do you get the images showing you, hey, it is staining, it is the polysorbate particles, but they strongly fluoresce. Now, if we use this same dye with different kinds of human IgG aggregates, and these were rotated IgG aggregates, you can see that they do not fluoresce under BODP30 at 21, and their fluorescent signature is well low in the background, right? So it is a dye that is specific to fatty acid particles and not to the more common protein aggregates. And as we keep moving forward, we can even potentially conduct co-staining of these lipids of BODP and THT for essentially multiplex detection. Here we have a mixture of fatty acid particles in IgG, where you can see the fatty acid signal corresponding to the lipid channel. This is the protein signal corresponding to the protein channel. And you can see how if we plot the lipid channel fluorescence versus the protein channel fluorescence, we get these two very well separated a distinct peaks for all of these different particles, right? We have like our fatty acid particle gate with thousands or more particles per mil, and the same thing for the proteinaceous particles. And we have the images to prove it. As you keep moving forward, this is a very sensitive assay, and we've heavily characterized and spent lots of time, and we have very low limits of detection down to 10 micromolar parts of lauric acid, and it's comparable in surpassing the existing assays in the literature, but we actually, at the end of the day, it is much faster, and you have the images to prove it. So it has very low LOD and very low LOQ in the presence of protein, and we have an application note that we'd be happy to share with you or talk to you in more details later in this webinar about this. But it's very specific, it's very sensitive, and it helps you understand and characterize these types of problems, right? So this is probably something that many of you have faced, in which, say, you take your samples out of the freezer or the fridge, and you might even say, hey, in this sample, I remember that I only have my excipients, there's some Polysorbate 20 here, but I don't even have any samples here. And then you put them in whichever tool you used before this one, and you try to measure it, and you measure something, but you don't really know what it is. Now, one of the things that it could very well be is degraded polysorbate fatty acid particles. In the case of Polysorbate 20, it could be palmitic or lauric or myristic acids. And here we see that only 3 minutes after thawing this previously frozen sample that was stored at -20, basically the particles start to fluoresce. As the temperature keeps, as the sample starts reaching room temp, the polysorbate starts to solubilize and fall back into solution, so there's less particles that are measured, and then after 20 minutes they disappear. So this is, of course, very much formulation dependent, but I'm sure that you've encountered something that looks like this, which is, hey, I saw these particles a second ago, but they're no longer there. And it's just because they can be highly soluble and again, very much sample dependent. And this is something you can quickly characterize in the aura, and because it's very fast, you can essentially capture where that polysorbate degradation is at one point in time. For some of these measurements that take a long period of time, you might potentially measure lots of particles at the beginning of a run and very few at the end of a run. So, okay, so that's just counting and looking at particles. As we move forward, let's look at some of these fatty acid mixes, right? So this is a particular fatty acid particulated sample made from myristic, palmitic, and lauric acid stored at 40 days in the fridge, and you can see how it forms these really beautiful particles that fluoresce very strongly under BODP 3821. And the longer you leave it in the fridge, they start forming more and more particles, where they potentially start aggregating and forming much larger granules. And you can see that not only from the images, but in the data, right? They have very strong fluorescence and larger particles, right? So much, much larger, and even to the axis of small, larger than potentially even 40, 50 microns. So the morphology and the counts and the shape for all of these different lipids, they change as a function of time. In this case of lauric acid by itself, it starts forming different kinds of particles over time, where it stands. You have these very small high contrast particles at the beginning. They start to become a little bit more translucent. They grow and start granulizing. They start separating, and then they essentially potentially you can see there's like a particle drop right between the 25 and 170 days, and then all the way down to very large clusters, right? So these are very polysorbate. One of the humbling experiences in working with polysorbate is that because of the sample dependence, protein dependence, uh, um, it's a very dynamic problem, and it's very specific to each one of these samples. But let me walk you through one of those examples. So for example, I showed you before how you can have formulations in which your fatty acid particles, your degraded polysorbate components, do not mix with your protein samples, and you have very nice discrete signals. But depending on your formulation, you might actually have not just two very well distinct clusters, which you still have there, but potentially more particles that fluoresce in both, right? And if you take a look at these images, and that's one of the advantages that this is not a blind method, right? You can go back and prove to yourself what's going on here. So if I were to click on this image, you would see an individual degraded polysorbate particle fluorescing. If I look at the protein channel, I would see in red like these very cool looking protein aggregate in the red. But as you go in towards these mixed samples, right, you can start saying that it's mostly these orange samples. So basically a combination of both of those, of proteins mixed with the polysorbate. But, you know, in some cases you have essentially independent particles where you have a protein aggregate front with a polysorbate fatty acid particle mix in the end. So it's incredibly high throughput and verifiable because you can look at these images and use them to characterize your process. So I hope you're excited about this as I am. Since we have a few more minutes, I'll show you a couple of more examples of how the aura can be used to tackle some very important problems in formulation development, particularly catered to some of these newer drugs. And in the case of gene therapies, you know, low volume analysis is very important because you're sample limited, and the traditional flow imaging techniques are essentially lacking because they don't enable you to characterize the stability of these samples, given their gas guzzling large volume requirements. But here you can see how we deploy the aura to characterize the physical stability of AAV of 3 different serotypes, 2, 5, and 8. And you can see that we have 2 stress conditions where with these gene therapies, they were not stressed in the top, but they were thermally stressed at the bottom. And just with 10 microliters of sample, right, you can see how basically AAV2 didn't produce a lot of particles when it wasn't stressed. AAV5 produced a few, but this AAV8 sample actually had quite a few particles even prior to stress. While with this significant 73C for 2-hour stress, AAV2 formed large discrete particles. AAV5 formed these long fibrils, while AAV8 formed this sort of small particle smear, if you will. So they all have their own, and to coin a word used by one of our collaborators, they all have their particle personalities when subjected to different stresses. When you stain them, you can basically prove that what you're seeing is not some feature of the system. These are when stained with a protein dye, they strong very fluorescent, showing that these are in fact capsid particles that are degrading. And while this is, of course, an extreme situation, in the rotated and the freeze-thaw cases of stress that we subject to these serotypes, they also show, particle formation and different kinds of particle formation, right? So we have some tall particles. The blue channel, as I showed you before, is I use it to characterize tall particles, but there's still like these red, proteinaceous particles, these capsid particles, and you can use this to characterize the stability of the samples in high throughput. Where the controls, if we just look for the sake of time at this AAV2 stress sample, the controls hold. There's very few particles, but even though there's still some there, when stressed, lots of particles are formed, and the rotation of the freeze-thaw samples formed more aggregates than the control, but not that much. And as we keep moving forward, I think one of the things that people have resorted to use, particularly for these gene therapies, as a substitute to conduct particle analysis. They've used low volume techniques like DSF and saying that the melting temperature is predictive of particle formation. And basically what we show here is that it is not. If we just focus on, there's this really nice paper from PharmaRes that characterizes a TM for a bunch of different serotypes. And looking just at, say, AAV2, everybody says, yeah, AAV2 is very famous for being unstable. And that is true. It's famous for having a very low TM. But, however, on a physical stability standpoint, AAV2 was actually the one that was least prone to particle formation, right? So I'm not saying the melting temperature is that important, but proteins unfolding as a function of temperature is very different than large aggregates forming. And before it was very difficult to measure the two, but this technology allows you to do that, so there's no reason not to. And we found a tremendous impact in this field and are excited to continue to support it. So because many of you came here interested in understanding polysorbate degradation, many of you are probably working with or getting samples from cell line development. This technology can be used to conduct particle analysis as early as cell line development and handle these complex samples without you worrying about the system clogging. One of the ways that we do that is that you probably have ELISAs used to characterize, the yields or the titers of your sample. Right? Maybe many of you here are probably experts in fluorescent labeling using ELISAs. And you can use your very well-established assays to characterize whether your product is degrading or if you're seeing some other stability failure mode. And in this case, using antibody labeling, we can see how human IgG particles, fluoresce under antibody labeling, while proteins in this case, that they were not antibodies, lysozyme particles. They fluoresce under a protein stain, but they did not show the same signal. So it is quite interesting. So we can expand this. Aura is a platform that is compatible with your existing assays and helps you characterize the physical stability of your product. And as deployed to cell line development, let's say that you have antibodies against your drug product, you might be able to you can use this technology to understand whether you are producing protein aggregates from the ground up. So I understand that in cell line development, basically you select for cell lines that produce the most antibodies, but those can actually be quite unstable. And we basically shown that antibody candidates have been potentially labeled as winners. This green label right here shows that these cell lines are secreting unstable antibodies, and you can quickly see not only you may have methods to qualify which one are the best producers, but now you can qualify which of those are actually producing viable product that you can push downstream and be confident that it's going to work and not aggregate. And of course, the negative controls hold. So what we're seeing here is a drug product aggregating. It's not a feature of the primary, the secondary antibody binding against itself. So one of the advantages of the low volume and the fact that you can clog these channels because it's not channel based is that you can handle complex samples. So Aura is a platform. And you can use it across very different workflows, whether you want to stain your samples in solution, to stain the samples after they have been filtered and measured on the membrane. There's advantages to each, but the good news is that because it's such a high throughput platform, the assay development can be conducted very quickly, and you can ask several questions about your samples, however you think is best fit. Certainly, we have assays that we go that we recommend and we go out the door. But as with all techniques, and particularly this is a platform that enables optimization of your workflows. So why Aura? As I tend to say, why not Aura? It is high throughput subvisible particle analysis for all biologics. It is an excellent tool for characterizing degraded polysorbate. It is sensitive. It is specific. And it can handle all sorts of different degraded fatty acid particle components. It's compatible with USP 788, and it is in a USP I788. It enables low and large volume analysis in high contrast, and it provides detailed information on the sizing, counting, the morphology, and the ID of the particles in question. It's compatible with most fluorescence and cell assays. It's easy to use. CFR 21 enabled. I do not like to read from a slide, so I'm actually looking forward to getting your questions, and hope you share your enthusiasm for the Aura as much as I do. Thank you so much for attending.
Hello everyone, it's Stephen again from Halo Labs. Thank you, Bernardo, for your presentation. And now we will jump into the Q&A session. So let's see here. The first question that we have is for when measuring fluorescence, how much extra time does that take?
Yeah, that's a good question. So the fluorescence measurement only adds about 5 to 10 seconds to the total acquisition because once the microscope has basically imaged and sized the particles in bright field where it is counted, basically then it's just excited in the fluorescence channel, which that whole process only takes a few seconds more than just counting things in black and white, if you will.
Is it possible to measure or quantify reading this wrong, undergraded PS80, polysorbate 80?
Let me take a look at that question, please. Is it possible to measure?
Or quantify undergraded PS80, polysorbate 80?
So this system is ideal for measuring fatty acid particle formation for different kinds of polysorbates, and I'd like to talk to the attendee in more detail to understand more. But I think the answer is yes.
Okay, non-degraded. Okay. Sorry about that. What concentration of dye is needed to sufficiently label the particles?
So very low concentrations of dye, actually. In this case, it's 10 micromolar BODIPI, and that is comparable to some of the concentrations used to, for example, stain, cells and the cell membrane. So that's, I think it's adipocytes, right? So I think, yeah, so basically it's even compatible with, liquid phase staining of cells, so very low concentrations.
So this is a two-part question. The first part, how much flexibility do you have in the fluorescence workflow?
Yeah, that's a good question. It's actually quite flexible. You can conduct membrane phase staining. That means you count and measure all of your particles after they've been filtered, and then apply a stain. Or you can apply a stain while the particles are in solution, which is what many people are more familiar with. I think they're both viable alternatives. I think the first one allows you to make sure that you don't have any potential false positives from dye particles being present. And the second one, I think, is common, and but they're both compatible with the system.
Okay. Actually, there's a lot more questions now coming about this topic. Is it necessary to mix the stain with the formulation?
Is it necessary to mix
the stain with the formulation? So in other words, I believe what the kid is asking is...
So sorry for speaking over you, Stephen. Yeah, no, it's not necessary, right? So the measurements that I showed here, they were all done staining on the membrane, right? So we quantified all of the particles, right? Particle size distribution, images, etc. And only after we conducted those measurements did we apply the BODIPI D3821. Right, so we made sure that the BODIPI didn't, that the solubility of the BODIPI, that the dye didn't interact, right, with the particles in the liquid. But and maybe for some samples you might want to do that, and still you could easily do this and essentially verify that those results are equal or better or compatible, just because again this is such a fast instrument and you can conduct both of those side by side. The experiments shown here, they were done on membrane phase staining, meaning that the samples were filtered and then the dye was applied on top of those to only ask the chemical question of, are these lipophilic particles or no?
Here's a nice follow-up question. Do you need to rinse after applying the different dyes?
That's a good question. So the short answer is because the particles have already been measured, right? So you are only working in the universe in which the particles have already been counted. You do not necessarily need to rinse the dyes after the fact, right? So the short answer is not necessarily, because your particles have already been counted. But again, it is adaptable to existing workflows.
Are you limited on the dyes you use, or can I do other things? For example, RNA-specific labels like Cyber Gold and Ribo Green.
That's a good question. Yeah, so the OR is a platform, and it's compatible with most protein and cell and nucleic acid assays. So I talked a little bit about antibody labeling. We can use common fluorophores like Alexa 430, Alexa 488. We have applications in cell therapy using DAPI stains. Here we're looking at both DIPI D3821. So it is compatible with probably most dyes that are out there, and also like in some cases we enable customization if you have something very specific in mind.
All right, let's just shift gears. We've got a lot of fluorescence questions actually. How does the sampling efficiency of Aura compare to MFI and FlowCam?
So I spent a few slides on that. So basically, FlowCam and MFI, they work in a microfluidic chamber, which it's hundreds of micrometers tall. There's different kinds, but let's say 300. The depth of field of the objective that they're used is about 4x. So basically, maybe it's a physicist answer, but only about 20 to 40 microns of that 300 micron tall channel is in focus, right? So 80% of those samples are not in focus. So I really don't understand how they claim 100% sampling efficiency just from basically the laws of physics. In our case, all of the samples are on the membrane in a singular plane, and all of the sample has been filtered through a well-defined physical constriction. So we characterize 100% of the samples in high contrast, while they are in focus into 10 to 20% of the sample on a flow cell. So I would say that they claim 80%, right? So I mean, I get it from a vendor. You can put anything you want on the spec sheet, but that is just nuts. I would think that their sampling efficiency is true accurate particle detection is very low. And ours is just physically, just because everything is in focus, everything is in high contrast. It's really high.
Okay. How does the particle count data for Aura compare to, say, a Hayek?
That's a good question. And there's been a couple of papers now in the literature. Hayek tends to be the least sensitive of the subvisible particle analysis techniques. So both MFI and Hayek produce higher counts than Hayek. MFI and FlowCam produce higher counts than Hayek. They're more sensitive, and we tend to be more sensitive than the flow imaging techniques. So we are more sensitive, and it is very important to do that because there have been, well-documented cases in which the presence of subvisible particles that were missed from just following the Hayek compendial method. The only differences in a sample in the case of Amontis was subvisible particles. And that resulted in unfortunately, in several patients dying from anaphylaxis. So yes, Hayek is the least sensitive of all methods. It is potentially one of the oldest ones, but is the least sensitive of all. And we're the most sensitive of the three methods.
Great. And there's a follow-up question to that, at least it relates to Aura capabilities. Is it method compatible with USB or is it validatable?
Yes, it is. So this is a USB 788 and membrane microscopy inspired method. Digital imaging methods are allowed in USB as per USB I788. So there's a direct pathway to validation.
We have a question. I believe this is relating to our fluorescence capabilities on the black plates. Are these the same plates used on the horizon?
Good questions. As far as fluorescence capabilities, no. So the fluorescent plates are used to just do particle imaging, counting, and sizing. These black plates that I briefly showed before, they can also do imaging, counting, and sizing, but because they're black, they have proprietary materials that enable them to support labeled workflows. Right, so you can use physical stains and labelings like the ones that I've described your different kinds of fluorescent stains. So no, they are not the same plates, and the horizon plates do not support fluorescence because they don't have this fluorescence suppression background behavior. And if you try to use the horizon plates, you would basically get a lot of native fluorescence from those white plates that would make these measurements impossible.
Okay. So what is the upper and lower end size limits for the Aura?
So we spec for particles greater than a micron and all the way as big as 5 millimeters in size, so about a well. So we've documented large fibers that are present. They can be pretty much as long as a well itself. So yeah, we cover the entire subvisible range.
What's worse, measuring fewer large particles or a lot of small ones?
I definitely think that large particles, right? Because you have few large particles, you may not pass USP 788, and those are more likely to cause, capillary occlusion and have more immunogenic effects. So you definitely want very few large particles.
Well, this one's a lengthy one. How do you deal with mixtures such as residual host cell protein? Can you label specific proteins like an ELISA type assay?
Yeah, we can label specific proteins like an ELISA type assay. Again, this is a physical stability measurement, right? So if you have monomeric solution, like maybe if your host cell protein's aggregated, yes, we could detect it. If your host cell proteins are in very low amounts and they're still monomeric, they would basically filter right through the membrane. So, yep, that's, yeah.
Okay, so we're jumping back to fluorescence topic. How do you ensure fluorescent dye particles are not confused with stained particles?
So one of the most common workflows recommended to use with this system is that you run it like you would in traditional BMI. That's you background the plate, you filter the sample, and then you measure it. So at that point, you characterize all of the particles that are there. Once those particles have been characterized, you then add the dye of choice to ask the chemical question you're interested in. And then you look for positive fluorescence signatures of the particles in question. So if they were dye particles, it's very likely that they wouldn't fall over like a particle that was already measured. So this staged approach really helps not confound dye particle effects, which it can happen at large dye concentrations, and if you have dyes that are, for example, insoluble, right? So it depends on the assay that you have in mind, and that's one of the reasons why, as a default, we do recommend going with a membrane phase staining approach.
And this is related to the concentration of the dye. What concentration of dye is needed to sufficiently label the particles?
Thank you for that. So very low, in the case of Bo-Dippy, as low as 10 micromolar. If you have highly water-soluble dyes, you can use lots of, if you really want to, for example, in the case of thioflavin T, we can use millimolar amounts of dye because it's highly specific and water-soluble. So it offers a big range, and I think what's most important is that as long as the dye is soluble, and you can characterize that with this system, it's actually how we do it, is we, for example, test how much dye can you run before inducing precipitation, right? So you can measure that very easily. So you can actually optimize and dial in the amount of dye that you think is needed with the system and actually characterize its physical stability in the process.
Great, thank you, Bernardo. We still have tons of other questions, but we're at the top of the hour. I'd like to thank you again, everyone who attended the webinar, and I'd like to turn things back over to Elizabeth to close us out.
Thank you very much, Stephen, and thank you for your presentation, Bernardo. I'd like to take this opportunity to thank Halo Labs for sponsoring today's event. Finally, I'd like to thank all of you for coming and spending this time with us. We're very grateful you chose to be here. We know you're being pulled many directions, and we're grateful for your time. So on behalf of Cambridge Health Tech Institute's Global Web Symposia Series, thank you all so very much. Stay safe and have a great rest of your day. Bye-bye.